So I would like to thank organizers who invite me here, Duncan in particular. I'm going to talk today not about glutamate receptors, about TRP channels, and we've already heard about them enough. Sorry. All right. So when we're outside, right, we actually know, like we look around, right, and we know cold and hot. But not only we know by looking, right, we actually feel it when we cold or hot. So what happens if we touch an object like which is hot? Our sensory neuron brings information from the touch place to the spinal cord, where it synapses to the motor neuron, which brings information back to cause reflex. But if we focus on the touch place, right, it has a lot of, our skin has a lot of ion channels which are sensitive to changes in temperature. What happens, these channels, in response to temperature changes, they open or close their pores. This depolarizes membrane and in turn generates action potential, which travels along the sensory neuron to the spinal cord and synapse to either motor neurons to generate reflex in the previous slide, or sends signals to the brain to generate more complex reactions. And I'm not going to talk about this part. I'm going to talk today about sensory part and ion channels in particular. So as many of you know, many temperature-sensitive channels belong to so-called TRP channel family. And representatives of this family are sensitive to temperatures in broad ranges from noxious cold to noxious heat. Interestingly, they also regulated by small molecules which in humans are associated with senses of smell and taste. And you know about these channels because recently two gentlemen, David Julius and Artem Potapatyan, were awarded Nobel Prize for discovering these ion channels. So what happens, for example, with heat-sensitive ion channels, which normally are silent, as you increase temperature, then at with certain temperature, they start to conduct current, and dependence of current on temperature is very sharp, and that causes high temperature coefficient, which is characteristic of temperature-sensitive ion channels. And similar with cold-sensitive channels, just as the temperature drops, they start to conduct current. I'm going to focus today on TRPV3, which are warm activated ion channels, sort of on the hot side. And sent to Jean yesterday, she gave a nice introduction, so I'll be very brief about what is TRPV3, mostly expressed in skin, also in other organs, including brain. Physiological functions are also correspondingly related to skin physiology and pathophysiology, also to long-term depression. And the pathology is related to a number of diseases, including chenilopathies. Single mutations can cause, in TRPV3, can cause different skin conditions, including this severe condition called Olsen syndrome, also other skin diseases. Interestingly, TRPV3 is also reported to be involved in colorectal cancer and skin tumors. So very important drug target. As I said before, as a member of TRPV3, TRPV family, it's activated by a number of small molecules, and here some of you can recognize the natural compounds which activate TRPV3, and also synthetic one. This is very common activator or inhibitor of TRP channels called 2-APB. This channel has very interesting specific property called sensitization, very different from majority of ion channels, which is in response to repetitive stimulus, this channel conducts increasing current. So the current amplitude increases. And this is opposite to majority of ion channels, which in response to repetitive stimulus usually reduce current, and we call this process desensitization or inactivation. And sensitization also happens not only in response to agonists, but also in response to heat. And here's impulses of heat repeating one after another, applied to whole cell HEK cell recorded in whole cell configuration. Recording is done by our by the lab of our collaborator Vicky Vlacheva. And you can appreciate first the small currents through TRPV3 gradually increase with amplitude. And this sensitization can be observed not only in response to repetitive simulation, but even if you increase temperature gradually, you start seeing more and more current conducted. And this is different type of recordings. Here, recordings are made in lipid bilayer, work done by our collaborators Ella Zacharian and her team. And here, several channels incorporated in a lipid bilayer start firing. You can see more and more current coming out. And this experiment can be done by introducing just a single channel into lipid bilayer. Here's the example. At 27 degrees, you essentially don't see any current, and then as temperature increases, you start see firing, which increases with temperature. The voltage-current relationship basically linear, which is very different for temperature dependence. It's very non-linear, and you can appreciate open probability stays close to 0 till about 38 degrees, after which it sharply increases. And this sharp increase in open probability causes this high temperature coefficient, the characteristic of temperature sensitive ion channels. So my lab is structural lab mostly, and so what we've done, all right, what these two gentlemen in particular, Kirill and Artur, postdocs in my lab, what they've done, they purified TRPV3, and then they used nanodisk protein and lipids to reconstitute this protein into lipid nanodisks. And then they applied to cryo-EM grid and put it into microscope, shine the beam of electrons in them, and they're getting these projection pictures, which we call micrographs. You can appreciate these little red circles actually show individual particles of TRPV3. If you average a bunch of them, then you start seeing what we call 2D class averages, and you can start appreciating even secondary structure features start to come out. And we've been lucky enough by using MSP2 nanodisk, we got reconstruction to resolution beyond 2 angstrom. And you can appreciate holes in aromatics, water molecules become visible. And this nice resolution obviously allows us to build a model, de novo. This is, many of you have seen these structures. Typical for vanilloid subfamily TRP channels. Includes the transmembrane domain, the core of the ion channel formed by 2 transmembrane helices and reentrant p-loop. On the periphery, 4 tetrahelical bundles, which are called S1, S4, or in voltage-gated channels, this is voltage sensing domain. The characteristic feature of vanilloid subfamily TRP channels, intracellular skirt, which mostly formed by ankyrin repeats and 3-stranded beta sheets, which are connecting the elements of the skirt. This is a steady picture of the same thing, right? And here's a topology of single subunit. So it starts with 6 ankyrin repeats, then followed by beta hairpin linker domain, which brings it to transmembrane domains, which are split into S1, S4, and pore domain, followed by this what we call TRP helix. The characteristic feature of TRP channel family, the helix runs nearly parallel to membrane, followed by C-terminal hook, and the last strand of the 3-stranded beta 3. Okay, so now we purified it, and we've been going after activation, watching activation of this channel in structure. And first we done control experiments, so you can see the temperature was held at 4 degrees, and when we did cryo-EM experiment, we got population of particles representing just single state, which I call closed. Now you can appreciate why, because the pore of this channel has 2 constrictions. This particular one at the bottom has hydrophobic isoleucine 674, which create narrow point and unlikely to conduct water ions. But to be sure, we collaborated with MD group. They ran simulations, so the group of Roman Yefremov. They ran simulations and clearly showed that nothing, no water, which is blue mesh here, no ions, which are red mesh here, go through this channel. Okay, so this is control. That's what we expected. Now, let's apply temperature, right? So when we apply temperature, we use that protein to carry on the cryo-EM experiment, and we found two populations of channels, two populations of particles, and I call them closed and sensitized. Why? You can see in this state, the pore becomes wider. And we've been so excited, we thought, well, we must have opened it. And then our collaborators disappointed us. They ran simulations. They showed at this point, isoleucine 674 still doesn't allow ions and water to go through. Well, then we've been looking for tricks, right? One trick which actually we've been using, we changed the type of nanodisk. So this is circular nanodisk called CNW11. So we performed experiment, control experiment at 4 degrees, got similar closed state, non-conducting, and then we applied set of stimuli, heat stimuli, and then subjected the protein to cryo-EM. And we also found two conformations. Now I call it closed and open, right? Why open? You can appreciate why, right? So the pore at this location become really wide. And now our collaborators really made us happy. They ran simulations. They confirmed that both water and ion go through this ion channel pore. So I showed you, actually, I was calling 4 different states at 2 different temperatures and 2 different nanodisks, closed one. And they're actually the same. So this is superposition of 4 subunits from 4 different closed channels. You can appreciate it the same. And the profile looks the same as well. So overall, by doing these experiments, we got 3 different conformations. We got closed, sensitized, open. They have increasing size of the pore, and 2 of them are non-conducting, and 1 of them truly conducting open state. Great, so we reached the goal. We used heat, and for the first time, we activated the channel by heat exclusively, and we got it open conducting conformation. Okay, so what's happening? So one thing I already put on this slide, you can see this pink and blue color fragments, right? These are actually N and C terminal, and you can appreciate how differently they behave. If you take a closer look, in a closed and cold state, we have C terminals coming out of the third beta strand, wraps around this 3-stranded beta sheet, and creates a glue between 2 neighboring subunits, so it lines the interface. What happens with heat activation? This C terminus completely unwraps and forms this short alpha helix with completely different type of interactions between neighboring subunits. Instead, what we start seeing another polypeptide sneaking through this cleft and making a new glue. And previously, actually, us and other people been noticing this polypeptide, but it was unclear what it is. Now, with the high resolution reconstruction we can clearly build every single residue in this polypeptide and confirm for the first time that this is N-terminus of the neighboring subunit. So we have this very interesting behavior upon heat activation. Another interesting behavior concerns lipids. And many of you have been talking about role of lipids in regulation of ion channels. So if you focus on transmembrane portion, and these are lipids representing one single subunit, you can see the upper leaflet in a cold and hot state pretty much looks similar. But if you look at the intracellular leaflet, there are clear differences. And especially this, what I labeled with letter V. You can see in a cold state, right, in a closed state, protein forms this nice crevice, and lipid, there is a lipid, fits this crevice perfectly, nicely. And then with heat activation, this crevice completely disappears, same as lipid dissociates. So V stands for vanilloid. So this is vanilloid site, which was first discovered in TRPV1. In TRPV1, in the APA state, it binds phosphodienoacetyl lipid. This site also binds agonists like capsaicin or RTX, or competitive antagonists like capsaicin or analgesics. So we've been wondering what kind of lipid we have, and you can see the black mesh actually shows our experimental density, and you can appreciate that the quality of this density is far worse than the surrounding protein I showed you before. And the lipid has sort of head and two-tail appearance, and so what we've done with our collaborators, we just took different models of lipids and inserted into this density, run molecular dynamic simulations to see which ones actually will look similar and which ones are gonna stay in a place. And amazingly, majority of lipids stayed in a place, so they didn't dissociate very quickly from the spot. It's unlikely cholesterol because appearance of this lipid is quite different, but all other head and two-tail lipids are good candidates. And so we concluded that the site is promiscuous and specificity is quite low, but this question really requires more work and attention, and so this is a kind of future studies. So overall, the conformational changes are happening pretty dramatic. So the skirt goes towards, upon heat activation, goes towards the membrane by about 5 angstrom. And if you look along the axis of four-fold rotational symmetry, it rotates by 8 degrees. So these dramatic changes, actually, we can follow them in what we call a conformational wave activated by temperature. And we can start, for example, from a spot where a vanilloid site lipid dissociates. Upon this dissociation, we have these strong local conformational changes in the secondary structure of the S2S3 linker, which actually changes helicity from its N to C terminal portion. This, in turn, the dissociation of lipids is also causing S4, S5 linker move towards the S1, S4 bundle. And the movement of S5, which is accordingly going to the same direction, is followed by S6, which is tightly bound. And S6, as we know, forms the gate of this channel. That's what actually opens it up. This dynamical movement of S6 is linked to TRP helix, which moves, includes the movement of the C-terminal hook, the linker domain, and that in turn is transmitted to three-stranded beta sheet. And then, as I showed you before, right, that's flip-flopping of N and C termini. So this is a conformational wave. Now, we also used a couple different ways of looking at the structural rearrangements. And here I showed you 3 different states we got through our structural work. And in principle, the sensitized state by conformation represents some intermediate between closed and open. And so essentially what we can do, we can split the motion going from closed to sensitized and then sensitized to open. And so in this representation, what we've done, we measured RMSD with a short window of 10 residues sliding through the entire structure. And this representation highlights very much local secondary structure changes. And you can appreciate, I already told you before, S2S3 linker getting highlighted and obviously N and C termini. Interestingly, so this is close to sensitized. This is presumed highly temperature sensitive transition, right? And we have these changes. In sensitized to open, which is presumably a weakly temperature sensitive, we have no such changes happening. And then we looked at the measure of structural movement, which characterizes these conformational states. And going from close to sensitized, again, so this translation is measured by superposing channels based on S1, S4 bundle. And you can appreciate, again, we highlighting the same regions which undergo secondary structure changes, S2S3 linker and C termini, but also this movement involves, for example, the anchoring repeat domain. So it moves as a rigid body, but as a result of it is involved in the conformational wave which I showed you before. Interestingly enough, a completely different character of structural movement is observed for sensitized to open. These changes, these movements are really localized to the gate area. That's where the channel opens up and conducts ions and water. And this is summarized here in kind of to appreciate the extent of movement in this movie. So this is transition from the closed to sensitized. You've seen these dramatic rearrangements in NNC termini. The open to sensitized, these are mainly the site of action. And while it's much less dramatic, it's very important because that's actually what opens up the channel, right, and gives the conductance. Same, we're looking along the axis of four-fold rotational symmetry. You see this formation of alpha helices, dramatic rotation of the entire skirt. Now this iris-like opening and then closure of the ion channel in the middle. That's what gives conductance. And then coming back, right, rotation of the entire skirt the opposite direction, and the channel becomes closed. So what I told you today, basically we got three different states, and hypothetically we can describe transition from closed to sensitized, which is highly temperature sensitive, presumably, and then sensitized to open. So the first one is accompanied by dissociation of lipid from the vanilloid side. Also, dramatic secondary structure changes in the S2, S3 linker, NNC termini, movement of the skirt, and then the opening transition really involves just opening of the ion channel gate. And I guess I will skip conclusions, so we have time for questions. At the end, I would like to acknowledge people who actually done the work. So these are Apo and Luke, who started the whole story with TRPV3. Kirill and Arthur, who are current postdocs in the lab, all other members of my lab. Our collaborators, teams of Vicky, Ella, and Roman, members of their teams. Cryo facilities who helped us to collect data, different funding sources, and I would like to thank you all for your attention and happy to answer questions.
Thank you, Alexander. Thank you very much for wonderful data with a beautiful structure and a beautiful story of the transition of the gating. Please.
Okay, so fantastic talk. I'm enthusiastic because I see that at the end, the voltage sensor is activating the pore, but the movement of the voltage sensor is induced by temperature by changing the position of S2, S3 linker and S4, S5 linker. So it's basically, if I understand correctly, there is this force that acts from the cytoplasmic side and is touching exactly the right positions. So that's why these channels, they have a voltage sensor domain. Do you agree with this?
Well, voltage sensor domain misses charges, right? So it doesn't really react to any voltage. But so the role of voltage sensor-like domain in temperature activation is important because we've seen these secondary structure changes in S2, S3 element. It's unclear, though, yet what really causes the changes. So this part of the protein might not be the driver of these changes. So that's still kind of...
But all the movement is towards the linkers, the S2, S3, and S4, S5.
That's right, that's right. And so as we describe this a wave, right, so includes several parts of the protein. Interestingly enough, something I didn't tell, right, over time, over a decade or even more, people did mutagenesis in different parts of a protein, right? And every time they would find mutation which affects temperature gating, they would say, oh, this is a temperature sensor, right? So now we can map all these mutations, but then the next group would come up with completely different mutation in completely different part of a protein and say, no, this is a temperature sensor. So now if you map these mutations, they're all gonna be on this on the conformational wave. So in a sense, what's going on, it's if wave propagates, right? At any time, at any point of the wave, if you stick an obstacle, you will stop the entire wave. Okay? Yes.
I have two questions. First, you were mentioning that the change in the nanodisk was solving your problem, but you didn't tell us what kind of different properties the successful nanodisks had. And the second question is, thermodynamically, this threading in of the N-terminus is a really loss of entropy. How do you explain this under the condition that you're adding heat?
So the first question, the nanodisk, right? So interpretation is, again, this is just interpretation. So we probably, with the CNW11, we just, or CNW30, we create a larger space, right? So we give, lipids freedom to move more freely, right? And restrain the conformation less. So that's, now the second, the entropy changes is grandiose, right? But also the enthalpy, as we measured from estimated from single channel recurrence. So yeah, we've been trying to do some calculations, right, on that. It's very hard because some portions of protein remain to be invisible, right? So I described you the structure. Structure covers most of the protein. Some parts are invisible, including actually in a closed state, we don't see N-terminus, which then binds and starts appearing, right? So from the thermodynamical perspective, this is a very difficult problem because we actually don't see the entire system. And that parts of the system carry energy. So this remains an open question. We work with the modelers and theoreticians. That's a non-trivial question. Yes.
Okay, we will have two short questions. Jamin. Fujiwara? Okay.
Yes.
And then Fujiwara.
So that's very interesting that you're talking about the conformational wave.
Yes.
I think that for multi-dependent gating, you can also say there is a conformational wave, right?
Yes.
But in the multi-dependent gating, there is a sensor where the wave starts. And in your case, that people always say, oh, here's the sensor, there's a sensor. But where does the wave start? Do you have any idea?
Well, we can, now as we have structures and we see what is moving, right? Now people are free to choose, right? And I can tell you my hypothesis, right? But in essence, all parts which are undergoing drastic changes, right? Including S2S3 linker, including NNC termini, and as well as actually lipids surrounding the protein, right? They could actually play a very important role in sensing. So again, because this is a wave, it's very hard to say, hey, what is generate the waves? As soon as it generates, like it propagates. And this is, I would say this is an open question. I have my preferences and we can talk about that, but yes, I wouldn't put my head right now and say, hey, this is a sensor. I think we should be, still investigating this.
Okay, Dr. Fujiwara, please.
Thank you. My question is almost the same to his second question, so please pass to the next.
Okay.
Okay, Alex, wonderful
Yeah.
talk. So, now TB3 is categorized as a worms activated. And your experiment, you raise the temperature from 25 to 42. Actually, we worm, it's activated, TB3 activated. On the other hand, the TB3 channel never exposed to the temperature. Some paper says it needs a temperature increase over the 50 degrees Celsius to get it sensitized. How it can be explained in your model?
Yeah, well, that's still kind of, that's an interesting question, right? Because, so what we've done in the protocol we use, right? So we can do different ways, right? We can, heat it up. So we chose the pulse protocol for pure biochemical reasons, because protein survives much better if you do pulse simulation, right? And you have to do like decent number of these pulses till, you sensitize. And essentially what you do, you populate the open state more and more, right? But even after we do that, we still, when we plunge freeze it, right? There is a reverse movement of closure, and you have population of both closed and open channels. So the hysteresis, right, of going to open and then close, it's fantastic question, and it requires little more kind of detailed structural work than what I showed, because this is by far kind of the first grandiose step, right, to just see, hey, like, can we really open the ion channel with heat? Yes, we can. But now, it will require the more detailed kind of parsing of conformation, intermediate states, seeing like what's really, in what succession the conformational changes happen. So it's, I think, fair to say we don't really understand this yet, like the detailed mechanism of this gating. So it's more work to do.
Okay, thank you very much for a wonderful talk. Let me join thanking Alexander.